talk to us today about from little things, big things grow. Extracellular vesicles and their miRNA cargo in retinal health, degeneration, and immune modulation. And just before you start, Riccardo, is that, I actually can't see your picture anymore, but I thought I saw Yvette sitting behind you before. Is that Yvette there?
Yeah, hello. You can see Yvette, and I actually have Adrian as well sitting in as well, who are the stars of the show.
Cool. So just for those people in the room here, Yvette, before going to ANU, was an honours student here. So we have a link and hands across the Tasman here. So anyway, over to you, Riccardo. Looking forward to your talk. Thank you. Fantastic.
Just making sure I'm not in presenter mode. Is that correct? And everyone can see me fine? Fantastic. Look, I'd like just to start with an acknowledge to country. So I'd like to acknowledge the traditional custodians of the land in which we are meeting. So we're in Canberra, so the traditional owners of the land are the Ngunnawal people, and pay my respects to the leaders past and present. I've actually titled this talk From Little Things, Big Things Grow for two reasons. Firstly, we're really interested in microRNA. Sorry, Randy, this is going to be a microRNA molecular talk. And interested in understanding how these can be utilized both for diagnostics and therapeutic purposes. And so the little things are the microRNA, but the little things are also the amazing early career researchers that I'm starting with, that are starting their careers and that have been incredibly influential in the work and how it develops. So that includes Yvette, who you just heard about, Adrian and Josh. And so I'll be leading you through some of their work that they've done. The EV story will come. It's going to come about 15 minutes into the talk. But I'd just like to give you a little bit of background on the work that we do. So we're at the Australian National University in Canberra. We largely work on a disease called age-related macular degeneration. You heard a little bit about that from Henry. We specifically work on a type of age-related macular degeneration called dry AMD. Dry AMD currently has no treatments. And basically by the time you visualize that there is presentations of pathology, it's probably too late and the disease is going to progress. There is a secondary disease type called wet AMD. This can be treated. It's largely a neovascular disease where you get bleeding of vessels that burst out into the retinal space, and that leads to very acute, rapid degeneration. These can be treated with anti-VEGFs that sort of soak up some of those vessels. So essentially, with the disease and the disease pathology, we're looking at roughly 200 million people globally who have some form of age-related macular degeneration. This is predicted to increase to somewhere between 288 million and 300 million by 2040. It could increase more with the incidences that we're finding out more about blue light and the toxicity that could actually have on the retina. And it accounts, dry AMD, for about 90% of all cases. So what we do is we actually study the photo-oxidative stress and the subsequent inflammatory effects of age-related macular degeneration. We think these are two big components of AMD. So we're not the only people that think about this way. So there was a paper published back in 2013 by and Batty. And what he did was really eloquently explain the different pathogenesis cycles of AMD. And if we go from the top down to the bottom, we can see that aging is sort of the #1 factor for the pathogenesis of AMD, makes sense. And then just sitting underneath that is photo-oxidative damage and some genetic factors. I won't be talking about any of the genetic factors that lead to microglia dysfunction, but I will be talking a little bit about DICER and the effect that DICER has, because DICER plays a role in microRNA biogenesis. So this is back in 2013. And at that point in time, microRNA and the role in retina and retinal function and structure hadn't really been looked at all. So to simplify what and Batty actually found, what he was suggesting was that oxidative stress led to increased inflammation, and that increased inflammation led to central vision loss. And what we are trying to do is we're trying to focus our energies, similar to what Henry was saying, on therapeutics and diagnostics that can stop or kick up that changes in inflammation state that might happen very early in the pathogenesis of disease. Just to give you a little bit of an insight into what the mechanism is that we study and what we think, how it all sort of unfolds in the retina. Just got a little bit of an animation here and I hope it plays. Randy, I'll look to you if you've got thumbs up to say that it's playing fine. Excellent. So light enters into the retina. It doesn't matter whether it's a human or a rodent retina, although the cell types might be the same. The essential mechanism is the same. So the cell types might be slightly different, sorry, but the mechanisms will be the same. So cones versus rods. As light comes in and scatters across the surface of the photoreceptors, which are the light absorbing cells of the retina, you get this increase in energy demands, increase in lipid peroxidation, and eventually increase in reactive oxygen species. And the retina is really well tuned for dealing with that, except over time what happens is some of the photoreceptors start to break down and you get these movement of microglial cells into the outer nuclear layer space or the photoreceptor layer, and they should never be there. Once they're in there, they cause havoc. They start killing cells off that were healthy, they start autophagy, and they start recruiting other cells into this environment. So what we think is that if we can stop those initial cells from going in there or going in there to do their normal homogeneous sort of functions, then we can protect against progressive inflammation and progressive degeneration. So that's what we work on. All right, so what are the roles of microRNA in retinal degenerations and why are we interested in them? Well, they're cool. They're just cool. They're these 21 nucleotide base pairs. They come off a gene backbone, develops the same as any other gene would actually come. So you have here a piece of DNA. It then makes this little hairpin structure that is exported out of the nucleus into the cytoplasm. It's then cleaved by that enzyme called DICER. And if you remember before, Jay and Batty alluded to that DICER enzyme as being important in the pathogenesis of AMD. It cleaves this microRNA, and when it cleaves it, it actually cleaves it into its active form. Now, this microRNA can then act as a sort of barcode scanner, scanning the cytoplasm, looking for matches. Once it actually finds a match, it either represses that signal or degrades it. And in humans, it's more repressing than degrading, right? So they're really cool little molecules and they're endogenously expressed. And for us, we've found that, and others have found that it's really abundant in the central nervous system, and they're really important for regulating neuroinflammation. There has been a rapid development of RNA biology into clinical trials and specifically microRNA. And in the middle of the pandemic, we can see that one of the potential vaccines that are being developed is an RNA vaccine. So we're showing for the first time probably the really true therapeutic benefit of understanding and studying RNA and microRNA. The small size make them really easy to transfect. We're talking 21 nucleotides, so they're very small. And they have these multiple benefits because they can bind to multiple pathways. In fact, a single microRNA can bind to, on average, about 200 different messenger RNAs. Now that's great because you can control and shut down whole pathways, but it's also a major therapeutic hurdle because you need to understand not just what target you want it to bind to, but all of the potential targets that it could bind to. They're postulated as potential treatments for neurodegenerative diseases, including AMD. And one in particular, miR-124, which will become the real hero of this story as we start to unfold it, we have been studying lots of. We've also found that it's essential for the development of retinal tissues, including RPE and photoreceptors. So the first little things, big things grow, person that's been working on this is Josh Chutan. And we published a paper a few years ago looking at miR-124 dysregulation. And this was really the results that led us to studying extracellular vesicles and why we wanted to. So one of the things we wanted to do really early on was understand miR-124 and where it was located within a human tissue. So just for those who aren't familiar with the retina, over on the left-hand side, we have a schematic. This shows the outer nuclear layer or photoreceptors, and I'll use those two terms interchangeably during this talk. You can see the microglial cells here, and these are the Muller cells. And those are the three cell types I really want you to understand. But basically, light comes down here, hits photoreceptors, sends a signal out through the internuclear layer and ganglion cells and out to the brain. All right, Muller cells are responsible for maintaining homeostasis, and they're a glial cell, so they're a non-neural cell. And your microglia are your resident immune cells. So those are the key cells to understand from this presentation. So what we did was we did an in situ hybridization, which is a technique that allows us to visualize in tissue the location of this microRNA. And when we looked at a normal 90-year-old human tissue in the retina, what we found was that there was a heavy expression in the photoreceptors and a scattering in the internuclear layer. But when we looked at AMD patients, that was completely gone. So what we had here was circumstantial evidence that suggested that the absence of miR-124 was correlated with photoreceptor loss or photoreceptor cell death. The scramble is just a control. So to tease this out a little bit more, what we did was we went to our photooxidative damage model. Now, this is a model we developed. It's a model that enables us to do central specific cell death within a rodent and allows us to look at the changes that happen as a consequence of that. It's photoreceptor specific cell death, similar to what happens in age-related macular degeneration. And then as a consequence of that degeneration, you get inflammatory cells that move in. And we can put them in for a period of time and then look at the post-exposure period as well, just to show you the cute little guys in action. Here's our light damage boxes that we developed. Here's the mice. These are 5 days into the actual experiment. They're not bothered by the light at all. And all that happens is it kills photoreceptors. So essentially we have a model that causes toxicity, huge amounts of oxidative stress. That oxidative stress leads to inflammation, and that gives us a model to decipher some of these molecular interactions we're interested in studying. Here is the histology that happens over time in the retina. So if you look at 3 days, we've got this big thinning of the outer nuclear layer. In red is tunnel labeling. So this is labeling cells that are undergoing apoptosis. And what you can see is you're getting a thinning of the outer nuclear layer, and you're getting cell death specifically only in the photoreceptors. And importantly, not just during the damage period, but also in the post-exposure period, which means that once damage is set up, it becomes sort of perpetual. Importantly for this work, what we noticed was that tunnels seemed to peak at around 5 days. But if you look at miR124 expression, it actually decreased at 5 days. So what we're seeing is similar to what we saw in the AMD human patients. A loss of miR124 was correlated with increased cell death. We then knew that we had a model that we could look at the visual representation. So we went back to in situ hybridization to have a look at where miR124 was actually expressed. And this is where we got that moment of, okay, something interesting is happening here. If you look even 1 day after exposure to light, this is before you see a big increase in tunnel, but what you're seeing as far as an expression profile is a change in the miR124 location from a control being largely photoreceptors to being redistributed within the inner nuclear layer. And what Josh concluded from that paper was it was being moved from neurons to a glia. And the glia that it was being moved to was a Muller cell, and Muller cells have been found to play a major role in modulating inflammation. What Josh found in that paper was that the movement seemed to bind to a particular molecule called CCL2, which we knew had these inflammatory processes, the chemokine. So the question was, well, we could look at this data in one of two ways. Is it a change in expression that's happening in the Muller cells? Well, that was unlikely because groups such as Tom Ray's and Stephanie Walls had shown that Muller cells don't actually produce much miR124. So therefore, the only other thing that we could come up with was that there had to be some form of dynamic movement of this miR124 from the photoreceptors going to the glial cells. And hence, we're here. All right, so extracellular vesicles was our primary sort of mode of transport, if you would, from neural to these glial cells. And so we started investigating them. And I'm not going to go into what extracellular vesicles are because I think this group knows them better than I do. The real stars are sitting next to me. So we've got a vet wolf here, and I'll turn the camera a little bit, and there's Adrian over there listening in. It's a weird world we live in where we now give presentations with our students and postdocs sitting next to it, but I love it. So we published a paper this year. It was the first time extracellular vesicle has actually been harnessed from retinal tissue. We were the first to be able to do it. And I think the community, the extracellular vesicle community here has been incredibly helpful, and HEVI has been incredibly helpful in helping us develop some of these techniques and technologies and enabling us to do this work. So I just wanted to publicly acknowledge all the effort that the group has done. So before I get on to the miR124 story, I want to show you what we did to make sure that we were doing everything properly and that there was a model and a mechanism in place where extracellular vesicles were actually playing a role. So the first thing we wanted to do was just look at the concentration of these EVs and see what happened in our retinal degeneration model. So we took animals, we just then collected EVs during these time points of 1, 3, and 5 days in this photooxidative damage model. And what we saw was a decrease of extracellular vesicles basically consistently. So as damage is increasing, we're getting a decreased number of extracellular vesicles. We looked at their sort of size distribution, and what we saw was roughly a similar sort of size distribution using the technique that we used, which was ultracentrifugation. But what you saw was a decrease in the bioavailability. So there was a reduced number of these extracellular vesicles. Seeing that there was this reduced number, we wanted to then dive into, well, if we artificially reduce them, do we see a same sort of result? So we did that, and we were able to see a decrease in these extracellular vesicles as well. So you can see the decreased numbers of them, both in the absence and presence of light. Right? The interesting thing was when we started to look at the retinal function, what we saw was very, very surprising to us. If we reduced the extracellular vesicle, we saw a reduced retinal function. So for those who have never seen an ERG before, I'll take you through this trace. So what happens is you take a probe and you put it on the front part of the eye. You then flash a bright light. When that bright light hits, it causes a potential to fire, or actually to stop firing, and you get this electrical response. This decrease in electrical response essentially translates to photoreceptor function, right? This is referred to as the A wave. The B wave then comes up here, and there's this second peak up in the top point, and that's your B wave. So you basically get your photoreceptor function in A wave and second order neurons in B wave. And in both cases, what we were seeing was reduced retinal function. We then did the same thing, but with photooxidative damage. I won't spend too much time on it, but basically you saw the same thing. So if you had damage and you stopped extracellular vesicle, you got even more of a decreased functional response. Having a look at both the cell death response as well as the inflammatory response, what you can see is that if we blocked GW, you got increased tunnel, which is photoreceptor specific cell death. You got a change in the number and activation state of the microglia, or sort of morphological state of the microglia, so where they were located and whether or not they were amoeboid or ramified. Change in the retinal thickness, specifically in the outer nuclear layer, the photoreceptor layer, not in either of the other two nuclear layers, so the inner nuclear layer or the ganglion cells. You've got a decrease in the photoreceptors and increased cell death. So that made us think that the bioavailability of these extracellular vesicles were really important in protecting cell death, maintaining homeostasis, and stopping inflammation. So what I said was at this point in time to the guys when they showed me these results was, oh, this is great. This is fantastic news. Let's take these extracellular vesicles and let's look at the most important thing in them. Let's look at the microRNA composition. They're going to have changed, right? They're going to have changed because there has to be a different signal that's being sent out. And so we did the microRNA analysis on them. And as you can see from this principal components analysis that shows control, which is dim read versus photooxidative damage, there was no difference, none whatsoever. They were very, very, very uniform. In fact, if you look at them and you look at the distribution, the amount of microRNA in each of them was pretty much the same, right? What you will notice though is miR-124 was in really high abundance in them. So our current theory is that maybe the composition inside the extracellular vesicles as far as microRNA are concerned don't change. And it could be that there are other components such as proteins, lipid metabolites that we need to look at, sugars that we need to look at to see whether or not they're changing and whether or not they're influencing the postcode, so the delivery system of where these extracellular vesicles need to move to. And that's something we're actively trying to find collaborations to work with. So if anyone's interested, please let us know. When we looked at the microRNA composition though, so the sort of pathways that would be influenced by these microRNA, it was pretty fascinating to find that it was our similar sort of friends. So interleukin-1 pathways, which are heavily involved in age-related macular degeneration, toll-like receptor pathways that influence inflammasomes, which have been found to be involved in AMD, chemokine signaling pathways, apoptosis, and specifically found in gene-related disease associations, such as diabetic retinopathy, of which there is a lot of similarities between AMD and diabetic retinopathy. So coming back to the miR-124 story that brought us here in the first place, the set of experiments we decided to do was to take that same profile and do the GW, but look at what happened to miR-124, whether or not it moved within the retina. And so we did that experiment, and as you can see over here on the right, you've got here the in situ hybridization, as we'd seen previously, in the photoreceptors. If we photooxidative damage over 5 days and just use the vesicle by itself, it moves. If we do it with GW, we saw very, very little expression in the inner nuclear layer. So I think this is pretty strong evidence to suggest that there is an extracellular vesicle role that's playing involved here. The question is why, and whether or not we can reverse engineer this to make some form of therapeutics. And that's something we're actively looking at. Just to give you a really quick snippet, and how am I doing for time, guys? Just talk. I'll keep talking. Tell me if I've gone over time. Really quickly, what we've done is we've tried. Plenty of time? Excellent. Yeah. So what we wanted to do was see whether or not we could use miR-124 specifically as a therapeutic. And this was in Josh's work. So can we supplement the miR-124 here at this time point and cause some form of protection? And we used in these set of experiments just a lipid-based vesicle. These were the initial experiments that Josh used. And so what we saw was a decrease in CCL2. We knew that that was a target partner of miR-124, so that was good. We saw a reduction of AP1, JUN. We saw an increase in photoreceptor rows. We saw a decrease in inflammation. And importantly, what we saw was an increase in retinal function as a consequence of supplementing miR-124. So now we think we're in a really strong position because we have these extracellular vesicles that we've got the microRNA composition for. What we want to do is take the top 10, 15 of those microRNA, see what combinations we can put them in to develop the best therapeutic. And with the top microRNA that we had in the extracellular vesicles thus far, we've already had a very promising effect. But what we wanted to do, and this is preliminary data, this hasn't been published yet, and we're still working on it, so please don't take this as absolute a word. But what we wanted to do was take retinal EVs and see if we could infuse them back into the animal, and whether or not we could replenish the depleted stocks that we had seen previously, and whether or not that had an effect. So what we did is we took 2 × 10⁹ retinal EVs that we had extracted from a healthy animal. We started the photooxidative damage. 2 days in, we gave the retinal extracellular vesicles delivered intravitreally. Then at 5 days, we did an assessment. And what we saw pretty much astounds us is that you get a really strong increase in the number of photoreceptor rows. You saw a massive decrease in inflammation and a small increase in retinal function. So this is very good preliminary data that we're building on now to see whether or not we can use retinal EVs as therapeutics and what are the best combination of molecules we can use to develop the best therapeutics. So just in summary, the presentation, we think microRNA stability appears to be important for maintaining retinal health. EV numbers decrease as a consequence of retinal degeneration, and retinal degenerations decrease microRNA availability in the retina. The decreased EVs and microRNA cargo bioavailability leads to altered retinal state and increased inflammation. And so what we think is happening from this schematic is that EVs are constantly traveling out, maintaining homeostasis. As a consequence of stress or damage, maybe there's a really big burst response of them. They get depleted really quickly. As they become depleted, you get further photoreceptor cell death, activation of microglia, and eventually progressive degeneration. One thing I didn't have time to talk about, and similar to what Henry said before, we're also looking at them as being diagnostic markers, and we're doing some work in the bloodstream where we're taking patients with age-related macular degeneration, taking their bloods, and looking for potential biomarkers in the blood from both EV and serum. So with that, I'd just like to thank the members of the lab, but very much so Josh, Yvette, and Adrian for all their hard work on this project. And I've put here Dr. Yvette Wolf because she has actually officially had her PhD last week, the week before, accepted, and graduation is in, when are you graduating? 2 weeks graduation. So it's officially in this format. Congratulate her to the doctor. I'd also like to thank the funding agencies, so the National Health and Medical Research Council, the Australian National University that gave me a fellowship a couple of years ago to work on this, ANU Connect Ventures, Retina Australia, Boots Foundation, and ORA for all of their support. And I'm happy to answer any questions you guys might have.
That was a brilliant presentation. Thank you very much, Riccardo, and congratulations to that. So while you're thinking, there's questions coming up already, but I'm going to use Chairman's privilege and ask Riccardo, I know it's standard practice to inject people in the eye in ophthalmology, but as a layperson and someone who might one day have to have that done to them, it sounds hideous. Is there any possibility that you could deliver extracellular vesicles, and you were delivering them on day 3. So disease is already well underway by day 3.
That's correct.
Is there any possibility that you can deliver them via a more peripheral route and actually have them target to the eye?
Larry, you've asked a really, really good question, something we have been actually talking about over the last couple of weeks. So yes, I mean, that is the holy grail. If you can deliver therapeutics in the bloodstream and have them end up in the retina, then you have a fantastic therapeutic potential. The biggest cost for treating age-related macular degeneration at the moment is that you have to go and visit an ophthalmologist to get the needle in the eye. If we could start treating it in the same way you treat diabetes, for instance, where a patient at home could administer extracellular vesicles with even current therapeutics on the market, so Lucentis or Avastin, and deliver them directly to the location, that would be great. That would work really well for wet AMD. For dry AMD, what we need is we need therapeutics that can target specific cell types in the retina. So you don't just have to get them into the retina, you then have to understand the postcode delivery system. So that's what we want to try and work on. We want to understand what is specific about the macula, what proteins might be specific to allow those extracellular vesicles to go and travel into the macula, and then understand and unpick that to see whether or not we can engineer the best possible therapeutics we can. So yes, that's exactly what we'd like to do.
Thanks. Questions, Moira?
Very nice talk. Just a simple question. It was really nice to see the IBA1 to decrease. What would be even more interesting is to look at the phenotypes. Do you also have a decrease in M1 and an increase in M2 with the population that's also decreased? Because from memory, MIR124 has been associated with changes in phenotype of microglia.
You're thinking along the same lines as we are, absolutely. So MIR124 has been shown in microglia cells to push the cells from what has traditionally been called an M1 profile, but I might refer to it as an activated profile, and an M2 profile, which is more of a resident homeostatic sort of form. And so MIR124 has been found more in that M2 profile. So we actually think what's happening is we're putting these extracellular vesicles or the synthetic lipid microRNA vesicles into the system. They're being picked up by the microglial cells, and that's causing them to become deactivated. And that would lead to a change in the profile. We have some really strong preliminary data to show that microRNA are absolutely involved in that way in our model. Watch this space.
Excellent talk. I'm curious, are you looking at the microRNA as a biomarker from the EV, or do you just looking at it as an extracellular RNA?
Yeah, so clinically, most of the time, the bloods are actually stored in one of two ways, serum or plasma. So we've been basically taking serum and extracting from that. But based on the results that we're seeing here, we think that there is a possibility of going back and reviewing and just taking extracellular vesicles as well. Because our theory is that they're communicating with other layers of the retina. They're probably communicating outside as well. But clinically, the storage of samples, as I said, is mostly serum, and that is our primary aim at this point in time.
Thank you. Other questions? Can I ask a technical one then? Your extracellular vesicles, when you're using them therapeutically, how old are they? Do you collect them fresh from the undiseased animal and then deliver them, or are you able to store the vesicles and still get an effect?
So we haven't tried storing them at all yet. And as I said at the start of this, we're very new to the extracellular vesicle space. So we wanted to do everything with best practice. And so we extract them fresh and then use them as fresh as possible. So pretty much on the day.
Right.
And I'll check with the bosses there that I said that right. Yeah, I got the thumbs up.
I often wonder, because when you're looking at therapeutics, which we think about with the placenta, you've got to actually, if you ever used a vesicle like that from a natural source, then we have to figure out some way of storing them and retaining bioactivity.
Yeah, you're absolutely right. And it'd be an interesting experiment to do of storing them for a couple of months and seeing whether or not they lose their effect. And I think that's something that we could definitely take a look at in the lab.
It was a really good talk, thank you. Is the idea of the biomarker that you pick up the AMD before it's obvious with an eye test? I mean, obviously you can see eyes decreasing or look at them. Is that you pick it up 5 years before and then apply the therapy? Is that the theory?
So we have a proposal in place at the moment to look at samples that have developed reticular pseudodrusen. So those that aren't in the eye field, reticular pseudodrusen is sort of a pre-biomarker that you can see in the back of the eye as a consequence of an indication of progressing towards degeneration. So we have samples from patients that were sampled about 5 years ago and have now developed retinal, developed this pathology. And what we want to actually do is look before the pathology actually eventuated, whether or not we can see any of these biomarkers. So there's been a lot of, in the literature, has been looked at the late stage. We want to do even before you can visually see any pathology developing. And that's our focus and intention at this point in time.
And presumably the therapeutic would be more useful at that early stage than waiting till it's too late.
That's exactly right. But if you can detect age-related macular degeneration earlier, maybe you could give antioxidants or anti-inflammatories at a much earlier time point before the disease starts, and that might slow down the progression of degeneration. AMD with ophthalmologists is often talked about. It's not a disease you have to cure, it's just a disease you have to slow down because death will take care of the eventuality, right? So if you can slow it down a little bit, you can allow an individual to have a better quality of life. And that's the angle that we're taking on this work.
Thank you.
Cheers, thanks for the question.
Okay, no more questions. That was a fantastic presentation, Riccardo. Thanks for joining us. It was early in the morning for you, I guess, at the start of the meeting. So thank you very much. It was brilliant.
My pleasure. Thank you so much, guys.
And now we're going to move to a different topic entirely. So we're going to leave the eye behind, and Dr. Augusto